Alrighty. Thank you guys for joining me today. My name is Rain Fasahi. I am a field application scientist at Diagnostic, and I'm very excited to talk to you today about some NGS foundations and also going through some of our portfolio, including Agilent, Amiga, and Element. Please make use of the Q&A section on Zoom. I do have colleagues on the call that will be answering your questions, and then we can also open it up to the floor at the end of this presentation. So today is actually part of a three-part webinar series. It is the first webinar in the series. The second webinar will be happening on the 11th of November by my colleague Rob, and he's going to be exploring NGS workflows across vendors. And then on the 25th of November, I have my colleague Ross, who's going to be presenting an introduction to NGS data analysis. The invites for these webinars have not gone out yet, but please just keep an eye on your email, as I'm sure they will be going out soon. So before I begin, I just wanted to show you where diagnostic fits in. We supply for a range of companies in the molecular space, all the way from sample prep right through to sequencing and interpretation. As you can see on the slide, we distribute for a number of big names in the market, such as Agilent and Twist, and we've recently started distributing for Amiga for all your extraction needs. And we're very excited to have Element Biosciences, so we have an NGS box, and then we distribute for Sequan and Hyrax for data analysis. So starting off, we are going to look at the evolution of sequencing. DNA sequencing has moved from first-generation sequencing, such as Sanger sequencing, which was quite slow and labor-intensive, and it was revolutionized with second-generation sequencing, or NGS, and this dramatically increased the throughput with short reads. And in more recent times, there is the third generation that's come about, and that's long-read sequencing, which allows for direct single-molecule sequencing of very long DNA fragments, providing more contiguous and accurate results. So you commonly hear NGS referred to as massively parallel sequencing, and this is due to the way that data is generated. During the sequencing reaction on board the instrument, millions of biochemical reactions occur simultaneously to produce sequencing data. And depending on that platform used, reads produced in a single run can be greater than 1 billion. The biggest difference is that genetic information can be simultaneously captured for multiple genes, compared to Sanger, in which each reaction captures information for only a single region. So the four main advantages of NGS over classical Sanger sequencing is the throughput. For Sanger, there's only one read that's taken at a time, whereas NGS allows for between 10 and 1,000 plus gigabases of DNA to be acquired during a single sequencing run. The cost, NGS is still an expensive technique, mostly due to reagents for running the instrument. However, due to the massive output, the relative cost per base, and therefore per sample, is significantly cheaper than Sanger sequencing. The input, less DNA is required to initiate the library prep, depending on the protocol and application. For some, you can start with as little as 1 nanogram of DNA input. And lastly, the accuracy. With NGS, genes and regions are often covered over 100 times, and these multiple data points for the same nucleotide give you confidence in variant calling. So quickly, just an overview of the NGS workflow. Sorry, I'm going to get my pointer. You start off with sample preparation. This can be from fresh, frozen FFPE samples, ranging from blood to tissue to environmental samples, really anything where high quality DNA or RNA can be purified from. And then you perform library preparation. This can either be automated or manual, and it's important to obviously use library prep kits that supports the platform that you will sequence on. Some of them are quite specific. So once you've performed library preparation, you'll end up with a library. that you will then load onto your sequencer of choice. So with certain sequences, like in MGI, you need a conversion protocol. Yeah, it's very dependent on what library prep kit you've used to, to determine the compatibility of the sequencer, but it's just an important thing to check. And then once you've sequenced your library, you'll end up needing to perform data analysis and reporting. A very important step during an NGS workflow is your quality control, which we will be going through in a bit more detail just now. So what is a library? A library is a collection of DNA fragments prepared for sequencing by performing a series of enzymatic steps. These steps are commonly referred to as library preparation. So you have your isolated DNA or RNA that's then sheared or fragmented, either mechanically or enzymatically. You then perform end repair, and this generates a blunt end fragments that you can add an A onto for A tailing to the 3' end of each strand. And then you perform adapter ligation. So adapters are short DNA oligos that contain the primer sites used by the sequencer to generate the sequencing read. They can also contain 6 to 8 base pair sequences called indexes or barcodes, and incorporating these barcodes allows different samples to be combined in the same sequencing run, so it allows us to multiplex. So you add those adapters on, and then you perform PCR. Using PCR primers complementary to the adapters, DNA fragments with properly ligated adapters are selected for and amplified, and you end up with multiple copies of your library. So following this exact method would produce a whole genome library, as there's been no target enrichment. This is something that in the beginning I used to get very confused about, so I like to spend some time here. But what is an index versus a barcode versus an adapter? So this picture is a basic structure of a prepared double-stranded DNA library. So on either end, you have your flow cell binding or P5, P7 platform-specific sequences, and this allows for the library to bind to the flow cell. And then we have our sample indexes, so your I5 and I7 sequences. These are also called barcodes, and these are short sequences specific to the given sample library, and it allows for multiplexing. Then we have our sequencer primer binding, and these are binding sites for the universal sequencing primers. And then lastly, in the middle, we have our insert. So that is our targeted target DNA RNA sequence from a given library. So no matter which NGS platform you use, you need to consider the type of library prep method used and which best suits your application. So the first example on the left, or number 1, is the easiest to prepare, it's less laborious, and that is with no target enrichment. So you would extract your sample, you perform library prep, and then you would sequence. However, this does have a higher cost of sequencing because it sequences everything. Your introns, it's not specific. Then number 2, we have target enrichment with probes, or otherwise known as hybrid capture. And this follows a method of sample extraction, library prep, hybrid capture, and then sequencing. And this target enrichment allows us to select the region of interest or genes to sequence. Then lastly, there's also a target enrichment protocol. However, this uses amplicons. So you would perform your extraction. There's a PCR amplification step, library prep, and then sequencing. So the choice between amplicon-based and hybridization capture-based NGS target enrichment, so 2 and 3, depends on the specific research goals, budget, and sample types. The amplicon-based methods are ideal for smaller panels and known variants, offering simplicity and cost-effectiveness. And in contrast, hybridization capture with probes is better suited for comprehensive studies requiring broader coverage and the ability to detect rare variants. So your no target enrichment can be used for whole genome sequencing, whole transcriptome analysis, and shotgun metagenomics. Some examples for hybrid capture include pathogen capture from mixed samples, whole exome sequencing, or custom gene panels, 50 plus genes, or targeted RNA-seq. And then for amplicon target enrichment, some examples include 16S RNA sequencing, TCR-BCR sequencing, or viral sequencing. So why perform target enrichment? It allows us to sequence only the desired regions of interest, for example, exons, gene panels, intergenic regions. It allows us to sequence more samples per run compared to whole genome sequencing, so there's higher multiplexing capabilities. There's smaller data sets, so it's less intensive computational analysis, which would result in a faster turnaround time. And it allows for increased confidence and accuracy in variant calls due to the higher depth of coverage in regions of interest. And it's more focused, so there's less variance of unknown significance. Most of the currently known driver mutations affect the coding sequence of protein coding genes. However, protein coding exons account for only approximately 1% of the human genome. So performing whole genome sequencing is not always cost and time effective. Okay, now getting into some nomenclature. Firstly, read length. So read length is fixed in short read platforms due to configuration of the instrument and sequencing chemistry. It's usually 100 to 150 base pair reads and can go up to about 300 base pairs. So the read length refers to the number of base pairs sequenced from a fragment. Long read systems such as Oxford Nanopore can achieve read lengths of over 1 megabase or 1 million base pairs. And it's important to consider the read length when designing an NGS experiment. Short reads are great for rare variant detection, but don't perform as well as long reads do for detecting complex structural variants. And there's also another of the factors that we need to consider, which we'll go through later on. It's just a little infographic to show the difference between short reads and long reads. So yeah, you need to piece together many more pieces to get your reads, whereas long reads, because they're longer, there's obviously less pieces you need to piece together. Then we have read depth or depth of coverage. So increased read depth improves variant detection sensitivity. So for diploid organisms, germline mutations require less coverage as the frequency should be close to a 50-50 split. It's usually between 45 and 55%. However, for somatic mutations, such as those in cancerous tissue, you do require a higher read depth, as the cancer-causing mutation can be in as little as 1% of the reads. So some NGS assays for cancer, such as liquid biopsies, require read depths of over 10,000x. So really, it's just how many times it sequences the region of interest. Then we have coverage. Coverage is referred to as the average number of reads that align to or cover the known reference bases. The sequencing coverage often determines whether variant discovery can be made with a certain degree of confidence at particular base positions. In a target enrichment assay, the coverage of a region is often reported as a percentage on target. Then we have coverage uniformity and on-target rate. So besides quality control of the raw sequencing reads, it's crucial to assess whether target enrichment has been successful. So for example, if most of the reads mapped on the target, and if the targeted base has reached sufficient read depth. So a low on target is lots of wasted sequencing data, and it signifies that there was an issue with the hybrid capture stringency or the design itself. And this leads to probes binding to off-target regions. Then we have coverage uniformity, which describes the read distribution along the target regions of the genome and refers to the uniformity of the target capture among the probe panel. It's hard to get 100% uniform depth of coverage, as certain regions, such as those with high GC content, can be difficult to capture and amplify. The uniform coverage reduces the amount of sequencing required, so to reach a sufficient depth of coverage for all regions of interest. As I said, it's difficult to reach 100% because of the GC content, and the low uniformity in some regions can lead to poor read depth or have been over-sequenced, so you have wasted data. As I just said, poor uniformity will lead to poor read depth in some regions. Therefore, the average read depth is not always the best metric to assess assay performance, as it doesn't give you an indication of areas with poor sequencing depth. Now, the commonly known Q30 score or Phred score. This is a sequencing quality score that measures the probability that a base is called incorrectly. So a Q score of 30 equals 1 in 1,000 base calls may be incorrect. So these days, short-read NGS platforms, such as Element, are closer to Q40 and Q50, providing ultra-high accuracy variant calling. So ultimately, the higher the Q score, the higher the accuracy of the base calls. Now getting into long versus short-read applications. Short read is more for targeted panels, SNP genotyping, rare variant detection, or whole transcriptome expression, whereas long read is generally for genome assembly or de novo genome assembly, detecting structural variants, mRNA isoform detection, and haplotype phasing. So what are the advantages and limitations of short versus long read? Long read, you have higher sequencing fidelity, you can sequence fragmented DNA, so FFPE. It's cheaper per gigabase and can generate higher coverage. It also allows for greater sample multiplexing and can perform RNA gene expression. The limitations of short-read sequencing is not able to fully resolve structural variants, phasing alleles, or distinguish highly homologous genomic regions, struggles with coverage of some repetitive regions, and it can involve more complex wet lab workflows such as hybrid capture. Long-read sequencing, the advantages, you're able to sequence regions that are difficult to characterize with short-read sequencing due to repeat sequences. You can resolve structural rearrangements and phasing of haplotypes. You are able to read through an entire RNA transcript to determine the specific isoform, and it assists with de novo genome assembly. It's limited by lower per read accuracy, limited availability of appropriate bioinformatic pipelines. It requires high molecular weight DNA, and it's not suitable for deep sequencing. So you want to sequence, which platform do you choose? Most sequencing applications are well suited for short read platforms, as it produces a large number of short reads at high quality, allowing for robust variant detection. And these data sets are widely applied to genomic studies of viruses, bacteria, mammals, and plants, with a wide range of input materials, including low input DNA, FFPE samples, and circulating cell-free DNA. However, certain analyses are improved by very long sequencing reads, up to several kilobases, And these are generated by our long-read sequences, such as PacBio, and the long reads are able to span large repeat regions, enabling near complete de novo assembly of small to medium-sized genomes with improved assembly of large complex genomes, such as plants and animals, and then more sensitive detection of large structural variants. They also are very beneficial for complex genomes that are highly repetitive in nature and have GC extremes. So, things to consider before you go off and start sequencing. What kind of sample am I using and how much do I have? Is it high quality gDNA from blood, cell culture, fresh frozen tissue, or is it degraded? Do you have micrograms, nanograms, or picograms? Is it RNA or is it DNA? What do I want to learn from the samples I prepare? You need to determine whether you want to identify SNPs or SNVs, indels, or more complex rearrangements such as translocations or inversions or copy number variations. Do you want to perform gene expression analysis? Do you want to assess biomarkers? Do you want to look at de novo genome assemblies? And something important to remember is one size doesn't fit all. Library preps and target enrichment technologies come in several flavors with their own sets of advantages and disadvantages. You need to know what to look for during your QC steps of a given library prep workflow. You need to do your homework and make a decision that meets your specific needs. And you also need to consider the computational requirements, such as hardware and bioinformatic expertise. Higher yields aren't necessarily better, especially when performing non-amplicon-based library preps. The less PCR, the better the results, and the higher the library complexity. Over-amplification can lead to poor performance. Set your expectations accordingly. Poor quality and very low input starting materials may require special handling. More input required with whole genome amplifications, and results from high quality gDNA are not going to be the same as results from FFPE gDNA. Lastly, don't be afraid to ask for help. While sequencing costs have come down, it's still not cheap. Reach out to your sequencing cores, other labs, or vendors for guidance. So now that we've gone over very basic NGS theory, I'm going to be taking it all the way back to the beginning of the NGS workflow, which is your nucleic acid isolation. And as I'm sure you've heard with NGS, garbage in equals garbage out, meaning you need to start with high quality samples. So this leads me to introduce Amigo Bio-tek to you. They have celebrated over 25 years delivering high performance DNA and RNA purification solutions with their manufacturing facility in Norcross, Georgia, America. They are ISO 13485 and 9001 certified for quality manufacturing that allows product traceability with rigorous quality and process controls. They also have a number of CE IVD marked kits, and most of their products offer performance advantages ranging from increased downstream sensitivity to faster processing times with a significant reduction in consumable costs. They have a really large portfolio that support a wide variety of samples, including whole blood, saliva, cell-free DNA, pathogen swabs, fecal, FFPE, plasmid, insects and mollusks, tissue, plants, and environmental kits. They have spin-based products as well as magnetic bead-based products, and 96 for silica plates. So here is some of their DNA kits. As I mentioned, they've got a huge portfolio, and they have a number of RNA kits as well. They also have an in-house automation team. So they have a database of scripts on the following instruments ready to go. However, the automation team is also happy to consult on any other processes, but for the processes on the screen, they have predetermined scripts already. They also have their own purification system. It is called the MAG binder FIT24 nucleic acid purification system, and efficiency, affordability, and precision and reliability is what it stands for. It's an all-in-one powerful instrument. There are two models available, one for research use and one for CE-IVD. And yeah, I would say it's comparable to a Maxwell extraction system. It's also magnetic based, and it can process between 1 and 24 samples at a time. And lastly, from Amiga, they also have an Ampere bead alternative called MagBind Total Pure NGS. There are no protocol changes against major competitors. It's a direct alternative to Ampere XP and RNA Clean XP. It allows for double or single sided size selection. It can be used for manual, or it's adaptable to most open-ended liquid handlers, and there's no programming or reprogramming of robotics required. So now that we have high quality starting material, we need to have a look at QC in an NGS workflow. So there are generally 4 parameters to measure when evaluating sample quality in an NGS workflow. One is to calculate the size distribution, or sizing. The next is to determine the concentration or the yield of the sample. We need to know if there's sufficient material for the downstream process. We also need to assess the integrity of the starting material. How intact is the DNA or RNA? It's important for workflows that require high molecular weight input material or high quality RNA. And lastly, we need to have a look at the purity. So in this sense, it's referring to the presence of unwanted nucleic acid contaminants, such as adapted dimers or other artifacts. Measuring these 4 parameters will guide you in your downstream processing of your sample. So for your input sample QC, we need to ensure that the integrity of our samples is good, or make provisions for samples that are not as good. But the important thing is that we need to know what the integrity of the sample is. So in this slide, I've just shown the different sample types, so DNA, genomic DNA, FFPE, RNA, and cell-free DNA, and the different quality or integrity assessments that we can perform on a number of different instruments. So all 4 of these instruments over here are Agilent instruments, and we do supply them. I'm going to be going into a little bit of detail about the tape station system, as it is our most commonly used QC system. But very briefly, we also have a fragment analyzer system. It's a capillary electrophoresis system, generally for higher throughput needs. And then we have a femtopulse system, which I will briefly touch on, but this is applicable for QC of long read data. And then lastly, we have a bioanalyzer system. This has been discontinued, but will be supported until 2030. So, as I mentioned, your input sample QC is one of the most important QC checkpoints, as you can already exclude samples from a workflow before spending time and money only to have a sample fail. All right, so the tape station instrument, there are 2 different tape stations. There is a 4150, which is this one here on the left, and then a 4200 tape station on the right. They do exactly the same thing. The only difference is the throughput. So the 4150 can process 1 to 16 samples at a time, and the 4200 can process 1 to 96 samples at a time. So these instruments are mainly used for NGS QC. It allows us to interrogate the integrity of our samples, and it gives us different integrity scores. So a DIN score for DNA, it stands for a DNA integrity number. So it's a score from 1 to 10 as to how intact your sample is. Then a RIN-E score, which is the RNA integrity number equivalent. The reason that the DIN score and it's RIN-E and not just a RIN score is that the RIN score was used for the bioanalyzer system and it's not calculated exactly the same way, so they couldn't give it the same name, but it is equivalent. It's still a score from 1 to 10 to determine how intact your RNA is. And then it also allows us to assess the integrity of cell-free DNA with that assay. Areas that aren't supported by the system include protein and small RNA. And the key features of the system is that there's scalable throughput. It's flexible to use. It's very easy to switch between assays. Fast results, it generally takes about 1 to 2 minutes per sample, or less than 90 minutes for 96 samples on the 4200. It's easy to use, and there's also a constant cost per sample as you use discrete screen tape lanes. And you can also reuse partially used screen tapes for up to 2 weeks. So as I've mentioned, these screen tape devices that are used in your tape station are easy to use. They are ready to go. There's no gel or gel dye preparation required. And you can also very easily and quickly switch between applications. So you could do a DNA run and straight after you could do an RNA run. There's a constant cost per sample by scalability. As I've mentioned, they have discrete screen tape lanes and you can use partially used screen tapes for up to 2 weeks. It's highly flexible. There's a broad DNA and RNA portfolio, which I'll be discussing in more detail shortly. Fast results, 1 to 2 minutes per sample, less than 20 minutes for 16 samples, and 90 minutes for 96 samples. There's a low sample need. Most of the assays use 1 microliter of sample. For high sensitivity assays, they use 2 microliters. And then there's zero carryover. So as we use discrete screen tape lanes and disposable pipette tips, it's 1 lane and 1 tip per sample. These are the different assays available. So we have our DNA assays in blue and our RNA assays in green. I'm not going to spend too much time here, but the most important things to look at when deciding on which assay to use is your sizing range. So you'll see each of these different assays have a different sizing range over here. And then your quantitative range at the bottom. So at what concentration will you be running your samples? It needs to fall within the quantitative range. You'll also see that we have D1000 assay and then a high sensitivity D1000, same for D5000 and RNA. The sizing ranges of these assays are exactly the same, but the quantitative ranges differs quite significantly. So when deciding on an assay, that is what you need to know. You need to know what size are you looking at, what size is your fragment or what size is your library, and at what concentration. Where would you or could you use a tape station in your NGS workflow? As we've gone through in quite a lot of detail, after genomic DNA extraction, you would want to have a look at the integrity of your starting material to determine whether it's worth running it all the way through an NGS workflow. You could also use it after fragmentation, after PCR enrichment, and finally, before you load your sequencer to look at the final library. So here is some examples of final library QC, specifically looking at size distribution. So this is examples of high quality NGS libraries run on 3 different Agilent systems. So our tape station, fragment analyzer, and bioanalyzer. And as you can see, it's a good quality smear. There's no artifacts or adapter dimers. There's no additional unwanted peaks. And with this, we can determine the size distribution of the final amplified library and also check that it falls within the recommended size for sequencing. So we've looked at the sizing of our final library QC, but something else that's very important is the concentration of our final library. And this we recommend using qPCR. The reason for this is it's the most accurate method to determine the amount of viable library, as it selectively quantifies the full length library fragments by using primers that anneal to the P7 and P5 and P7 sequences. So this means that it quantifies only fragments that have been prepared correctly. Yeah. Once you have become more familiar with your workflow, qubit can be used, but in the beginning, qPCR is the most accurate. And the reason we need our sizing and concentration to be accurate is in order for us to calculate our library molarity. So it's really important that we size our libraries properly and that we have the correct concentration, because we need both the concentration and the size of the library to calculate the molarity or the molar concentration of the library. And this is critical in NGS as it ensures that there's an even read depth across the pooled samples. It prevents overloading or underloading of the sequencer. So if you get your molarity correct, and you have good quality library prep, it ultimately guarantees high quality, consistent, and reproducible results. So by standardizing the molarity of each library before pooling, you ensure that all your samples are represented by the same number of molecules, allowing them to cluster and generate reads equally. So sizing and quantification is extremely important, and your best methods of doing that is using one of the Agilent systems for sizing, so tape station, bioanalyzer, or fragment analyzer, and then qPCR for quantification. And once you're confident, you can perhaps move over to a qubit. And as I mentioned at the beginning of the QC section was the femtopulse, and I said that that would be used for long read NGS. So when using PacBio or ONT or any other long read NGS system, you need highly intact high molecular weight DNA as input and into the shearing step, as the final expected read size can easily exceed 20 kb. So the high molecular weight samples are sheared to the target fragment size, and then an optional size selection step can be performed to ensure fragments are above the desired size cutoff. The femtopulse system is very beneficial here, as it offers an excellent solution for analyzing high molecular weight DNA samples with high accuracy. It utilizes a pulsed field electrophoresis technology, and that allows for precise quantification and qualification of DNA samples up to 165 kb. So this is quite crucial for evaluating the quality of long-read sequencing libraries and predicting the average read length in these long-read libraries. The femtopulse system can be utilized throughout the workflow to determine the integrity and average smear size of the input gDNA or sheared DNA, and then the size selected sheared gDNA. And now, what I think, or at least hope, you've all been waiting for is the introduction to the Element AVITI system. Element, sorry, my slides aren't moving. So you may not have heard about Element before, but they have actually been around since 2017. It was founded by 3 ex-Illumina and PacBio staff members, and they had the common goal of making sequencing more accessible by making it more affordable. And in order to do this, um, Can you see my screen?
We can still see you.
Okay, great. Sorry, well, don't know what happened there. My screen just went black. Anyway, as I was saying, um, so in order to do this, they invented their own sequencing chemistry, and this has resulted in affordable, high-quality sequencing data that I will show you today. So, as I said, they launched in 2017. However, they kept under wraps until 2022, and that's when they released their sequencing box, the AVITI. In 2023, they started their global distribution, and at the end of 2023, they celebrated their 100th install of AVITI globally. 2024 came with many updates to their chemistry, and halfway through the year, they installed the 200 system. Starting this year in 2025, they installed their 300 system, and they currently have around 400 systems installed globally, and we have 3 of those in South Africa. All right, so over here you can see the system. It's actually a dual-sided system. It has 2 sides. So you can load 2 flow cells and 2 reagent cartridges, and you have 2-way space. And these 2 sides can be run concurrently, or they can be run completely independently. So this just demonstrates the flexibility of the system. Essentially, you can see it as 2 sequences that are connected. Some of the basic features of the system includes exceptionally high quality. This is demonstrated by the high Q30 score. The spec of our kits is rated at more than 90% of the data will be at Q30 or higher. However, I will show you some real life data from our customers, and that will show you that the system can actually significantly exceed that quality score. When it comes to cost, AVITI is currently the cheapest per gigabase sequencer in the country, and we can get as low as about 180 Rand per gigabase. And that's a great way to do a head-to-head comparison to determine that what you're actually paying to generate data. So each of the flow cells can generate up to 2 million paired end reads, and we have a range of different kits with different read lengths, which I'll show you shortly. As I've mentioned, they designed the system with flexibility in mind. So I've already mentioned it's a dual-sided instrument, but the flow cells have also been designed to be dual-sided in a sense by allowing for individually addressable lanes. So um, this can happen by the press of a button. It is a paid feature though, but the flow cell is divided into 2 lanes. So if you were to set up a normal run, it floods the flow cell over both lanes. However, if you use individually addressable lanes feature by just hitting a button on the box, you'll load your 2 libraries into 2 different positions on your reagent cartridge, and your one library will flood lane 1, and then your other library will flood lane 2. This can be used for a number of different things, such as optimizing the loading concentration of a new library prep kit. You can load the same library at 2 different concentrations, and after one run using the 2 different lanes, you can determine the best loading concentration. Or, for example, you only have 16 unique barcodes, but you have 32 samples. You obviously can't sequence those together. With this feature, you can barcode 1 to 16 and put it in lane 1, and then you can barcode the other 16 samples with the same barcodes and put it in lane 2. So effectively, you're splitting the indices across the flow cell. Here are the flow cells that we currently have. 2 by 75, 2 by 150, and 2 by 300. But it's rather best to look at it as cycles, so 150 cycles, 300 and 600 cycles, as you can set up the run however you like in terms of your read lengths. We have low, medium, and high output kits. And we actually have three different AVITI models. We have the low AVITI LT or low throughput, and these can only run the low and medium output flow cells indicated by the blue dot. And then we have the AVITI, which is compatible with all the flow cells indicated by the green dot. And then we have the VT24, which I'll be discussing later on. However, it's compatible with all the flow cell configurations on the slide as well. And while we're here, I also just wanted to mention that the VT low throughput or the LT box is also upgradable to a normal VT. So this extended family of cloud break flow cells, that is the VT's name for their flow cells is cloud break. They deliver cost savings when you don't need a full capacity of a 1 billion read flow cell. And it allows for sequencing without batching from 250 million to 2 billion reads per run, with additional flexibility offered by the individually addressable lanes on the flow cell. And this ultimately will allow you to improve turnaround times on smaller projects with lower cost per run options, or minimize per sample costs by multiplexing on higher throughput flow cells. So when I introduced Element, I mentioned to you that they invented their own sequencing chemistry. And this is referred to as avidity base chemistry, or ABC. So if you are familiar with Illumina, you'll know what clusters are, and that is used to generate signal to amplify the library using PCR. Element is a very different way of doing it. So instead of a cluster, we have something called a colony. And what it really is a condensed ball on the flow cell that has been amplified using rolling circle amplification. And during with rolling circle amplification, we make around 1,000 copies on the flow cell without onboard PCR. So these copies are only made from the original template, and this eliminates compounded PCR error and improves sequencing accuracy. It also reduces artifacts such as index hopping and optical duplicates. And then after amplification, each colony is a continuous DNA strand that collapses into a ball with many sequencing start sites in close proximity. The sequencing chemistry, or the avidity sequencing chemistry, combines several innovations to reduce sequencing costs, all while improving accuracy. And one example of that is the 100-fold reduction in reporting nucleotide concentration required for base detection through the core chemistry of an avidite. So an avidite is a core fluorescence molecule with between 3 to 8 appendages coming off of them. This can be seen in figure B. And then there are 4 different color avidites seen in figure C. So one for each nucleotide. So green is T, and on the end of each arm, you'll see they each have a T. And this allows for multivalent bonding. So one avidite can bind up to 8 different primer sites, which is how they have reduced the concentration required to make a base core. So going back to picture B or figure B, it's showing you a single avidite bound to a colony. The surface primer is shown in blue and sequencing primers in red. So each arm of the avidite connects to the core of the avidite, and that contains the fluorophores, which attaches to the nucleotide straight. Then at the top, figure A, this demonstrates one sequencing cycle. There is a nice preprint that goes into the detail about the chemistry that I can share with you. But ultimately, a polyny is bound to the flow cell, which is indicated by the flow cell attachment oligo within the library ball. And then We have the sequencing primer sites, so the AVITI binds to an open base on a template at a primer binding site, facilitated by polymerase. We then wash off the unbound AVITIs. We then use fluorescence to detect which base is at that site. And then we need to step forward into the next position. So we remove the AVITIs and we block that position using a reversibly terminated nucleotide with a polymerase, allowing the polymerase to jump one base forward. We then remove the block and then we start again by binding another AVITI throughout the different cycles. So this separates the process of stepping along the DNA template from the process of identifying each nucleotide, allowing each to be optimized for quality and reagent consumption. Now going back to quality, this is some independent data from the University of Tokyo, and they are looking at the Q score over or per cycle, and they're comparing Illumina and NGI, which is in yellow and green over here, and then AVITI Cloudbreak, which is the two blues, and then AVITI Ultra Q, which is the red and the orange. This is I'm going to go into a bit more detail just now, but it's also an Element product that allows for Q scores of up to Q50 or up to at least 70% Q50 data. But there's a specific workflow you need to follow, which I'll discuss just now. But the Q50 data ultimately means there's one error in 100,000 base calls. So yeah, you can see even with the normal Cloudbreak, Element Cloudbreak chemistry, they're sitting with an average Q score of between Q40 and Q50 throughout the run. So why does this Q score matter? When it's compared to other benchtop platforms, we can clearly see improvements in the read quality, index hopping, and optical duplicates, and performance of homopolymer-rich regions of the genome, so long stretches with the same nucleotide. And this quality affects nearly every NGS application. So the reason Element is able to achieve such high quality is due to the rolling circle amplification instead of PCR to generate the polymers. They have random flow cells and their specific sequencing and surface chemistry. So now I want to show you some real data. This is from one of our customers in South Africa that are running exomes. And as we can see over here, the kit specs, it's a 2x150 low output flow cell, whereby we expect to get around 500 million paired end reads, which is around 75 gigabases of data. And what we can see from their run specs is they were easily able to achieve 10% more than that. They had 550 million paired end reads of output, which equated to 81.8 gigs of data. They had a Q30 score of 94.6%, but the mean Q score is actually 42.7. And we can see that this is demonstrated by the red line going across all the cycles of the read. So this just goes to show that you can easily max out your AVITI kits, exceeding the kit output without affecting your quality. This next case is another example from a customer in South Africa. It's a low diversity library. And what we can see in this graph over here is the nucleotide composition throughout the cycles of the run. So we have some base calls that are almost 100%, meaning that every base at that cycle is the exact same nucleotide. And a lot of NGS sequences really struggle with this, as you're flooding the flow cell with the same color. But AVITI's chemistry handles this really well. So, and I'll get into why just now. But this customer used a 2x300 medium output kit. They anticipated to get 200 million paired end reads with 60 gigs of output. For this run, they used something called expert mode, and that's also just at a click of a button on the box. And by doing this, you can increase the output by up to 70%. How they do this is it slightly reduces the quality filters in the polyny generation step. So it allows more filters through, and we do anticipate to see a slight quality dip of 1 to 2% in the Q30 score. But look at what we were able to achieve with this run. 334 million paired end reads, so 67% higher output than what the kit is designed to do. They had 100.2 gigs of output, and the Q30 score is still 93.44 with the mean Q score of 42. So it virtually hasn't affected our quality score at all. And they were able to get significantly more output. But the most important thing that even with these low diversity libraries is the fact that we only need around 2%, or this run was done with only 2% Phi-X. Whereas with other systems, you require up to 20, 30, 40% Phi-X, and that enables the other systems to do the color balancing. For those that don't know, Phi-X is a sequencing control. You're not using it for your samples, so effectively the output of the Phi-X is useless. But yeah, so you can't do anything with the Phi-X data that you generate. So with 2% Phi-X used, you are using more of the flow cell for your samples, and you can still max out your reads and use much less Phi-X and still get really high quality sequencing on low diversity libraries. So I'm now coming back to Ultra Q, which I mentioned with the Q50 scores. So what Element have done, they have combined advances in sequencing chemistry with their own library prep to address the most abundant sources of residual error in NGS data. So specifically, library prep error is driven by deamination and end repair, which are mitigated with this kit with the uracil enzymatic digestion treatment and dark cycling through the error-prone initial 15 bases of rig 2. So in order to achieve this Q50 or greater than 70% Q50, you need to prepare libraries using Element Elevate Mechanical Library Prep Kit. You need to use the Elevate Long UDI adapter set. You need to follow the PCR-free workflow. Your recommended input is between 200 and 1,000 nanograms, and the target average insert size is 350 base pairs. And they've pretty much guaranteed that if you follow all of these steps, you can get up to 70% of Q50 data. One headache with new sequencing platforms is that sometimes the libraries are incompatible with the new sequencer or may require a conversion. So for example, Illumina libraries, you need to convert before you can sequence on MGI, but that is not the case for AVITI. With their Cloudbreak freestyle sequencing chemistry, it directly supports more than 95% of libraries without the need for conversion, which saves time and money. So this means you can take your standard Illumina compatible libraries and easily sequence them on an AVITI with no extra steps. Right, so Illumina really was instrumental in the NGS world. However, in order for them to decrease their cost per gigabase, they released instruments with larger outputs, such as your NovaSeq. And in doing so, they were able to significantly reduce the cost of sequencing. However, it requires you to run huge numbers or volumes of samples at a given time. So sometimes you need to run thousands of samples. And what we've seen is that some labs really struggle to fill these runs. So they have the instruments that can perform the cheap sequencing, but it takes months in order for them to batch the right amount of samples to achieve that low cost. So sometimes the high throughput is not just a feature, but it creates problems for smaller labs. So we noticed that in the local market, there was a need for a medium throughput sequencer that can deliver running costs similar to production scale or ultra high throughput instruments such as the NovaSeq. And that's really where AVITI fits in. You don't need to run thousands of samples at a time in order to achieve the same cost per gigabase. It also has there's less capital investments, which means that institutes with smaller budgets can now offer sequencing at a similar or lower cost compared to the NovaSeq, while retaining the sequencing quality, if not exceeding the sequencing quality. Something else that Element has promised is they have promised that for the lifetime of your sequencer, they have a reagent price guarantee, meaning they will not increase the price of the reagents for the lifetime of your system. So as a distributor, we just have to note that obviously exchange rates and freight may change, but the base cost will not change from Element. Very briefly, I just wanted to show you this poster that Element released last year. It's a head-to-head comparison of the AVITI and the NextSeq 2000, looking at 16S RNA-seq. And the big takeaway of this poster, and I have already emphasized it, is that in order to do this, they used on the Element less than 5% Phi-X, whereas the same run on Illumina needed around 40% Phi-X. So you're effectively losing 40% of your data to the control. And then to cap it off on AVITI, along with their principles of making things super user-friendly and accessible, they've incorporated that with your data as well. So they have many options on ways you can handle your analysis and data storage. The first one is a paid service, LM Biocatalyst, and this is run through Amazon Web Services. The next is pipelines through Analysis Network. So if you're already using a partner such as DNA Nexus or GeneCode, we can set it up to stream directly to them. Or you have a fully customizable option. So if you have your own Amazon Web Services or Google Cloud, or if you want to run it locally, you are also able to do that. And now I mentioned that I would touch on it, the AVITI 24. So this was only released in December last year. And this is a 5D multi-omic system. It's truly the first and only fully integrated single-cell multi-omics platform. So it's an NGS sequencer with added capacity to do cytoprofiling. So AVITI 24 really does redefine what's possible with simultaneous detection of RNA morphology, protein, and their dynamic responses with spatial resolution. And additionally, because they've increased the processing power of the sequencer, all the AVITI kits we went through previously get an extra 50% of output without changing the price. So your sequencing gets even cheaper. Just touching on the multi-omic aspect, over the last 20 years, next generation sequencing has catalyzed an entire ecosystem of multi-omic tools, from bulk RNA-seq and single-cell RNA-seq to proteomics. And often these methods barcode different analytes, so DNA, RNA, protein. You then perform library prep and you sequence on an NGS instrument. But these traditional multi-omic approaches often rely on multiple platforms and complex workflows. But AVITI 24 allows you to interrogate at a deeper level with more integrated insights, and it gives you a full picture of the biology of your sample with one instrument. So while this ecosystem of multi-omic tools have been extremely powerful, the challenges remain with long workflows, multiple pieces of expensive equipment required, high cost, and unpaired measurements across different assays and samples, introducing additional costs and requiring stitching together different data types. Whereas on the AVITI 24, it's one system and you can get all of it out at the same time. So, as I mentioned, you're able to assess cell morphology. They're continuously working on the chemistry, but currently you can use the 6-marker AVITI cell paint, so you can have a look at cell membrane, the nucleus, actin, ER, mitochondria, and Golgi apparatus. And they have 350 targets for RNA detection, and you can also interrogate 138 surface intercellular and phosphorylated proteins. And then using the built-in cell segmentation algorithms, you can confidently assign RNA and protein targets to these single cells. And all of this is co-detected in a single run from the same sample in less than 24 hours. So this talk was not about a VD24, but I couldn't give up the opportunity to at least introduce it. Ultimately, it's the first benchtop platform that enables high quality, affordable sequencing and in situ multiomics in one powerful system. So you can sequence exomes on the one side to performing spatial proteomics on the other side on the same day. A VT24 really empowers endless possibilities for biological discovery in a single platform. If you would like some more information, there is a Element of VT24 webinar series that you can have a look at, or you are more than welcome to contact us and we can share more information. But I am now finishing off, just reminding everyone that this was part 1 of our 3-part webinar series. So please do join us for part 2 and 3 on the 11th and 25th of November. And that is all from me. Thank you very much for your time. I'm happy to open the floor up if anyone has any questions. No questions? Then we are done for the day. Thank you very much, everyone, for your time. I hope you learned something. I hope you enjoyed the talk, and I hope to see you at part 2 and 3 of this series.